Cells are the basic building blocks for all living things. They provide structure for the body, they take nutrients from food, they convert those nutrients into energy, and they carry out specialized functions. Cells also contain the body's hereditary material and can make copies of themselves. Mitosis is a type of cell division in which a cell is split into two cells that are genetically identical. This type of division is used in all the body: hair, nails, skin. Meanwhile, meiosis is a little bit different because it only occurs on sexual cells. Meiosis is a special type of cell division necessary for sexual reproduction. The cells produced by meiosis are gametes. In many organisms, including animals and plants, gametes are called sperm and egg cells. Well, the objective of this lab was to dye some animal and some plant cells. For this we used some containers, some alcohol, some laminates, some swabs, and we use the dyeing stuffs that were methylene blue, as you can see, and we also used lugol, that's a powerful dehydrate dye. Okay, now the teacher is putting lugol and blue, methylene blue on the containers. Then he's going to explain us how to do it. So he began by taking a little and thin layer from the onion. This is going to be the plant cell. We have to cut it and put them 30 seconds on the lugol and the methylene blue. Okay, after 30 seconds of dyeing on lugol, we can see the plant cells. You can see that they are very organized. They look more or less like trapezoids. They have some dots inside and that they have a very thick cell wall. But I want to explain you what's that ball, that circle that's on the cell. That's the nucleus. The nucleus is a very important part of the cell because the nucleus controls all the cell. It contains all the hereditary information and it controls the cell growth and reproduction. Now, if you get closer, you can see that the nucleus have a dot inside. That dot is the nucleolus. The nucleolus is very important part for the makeup of proteins and the ribosome. The ribosome is the one that produces those proteins. Although we saw pretty cool stuffs from the cells, I couldn't see any division, so that's kind of sad. But we continue with animal cells, so we start taking saliva from our cheek, then we put it on the slide, then we put the slide on fire so it will heat and the cells and the saliva won't move, and it will be easy to put the methylene blue. But unfortunately we put not enough, so the cells couldn't take enough and it didn't dye well, so we couldn't see anything.